Electron micrograph of a replication bubble in humans. Replication bubble? Huh? This is a strand of DNA that's been opened up so that it can be replicated. Remember that DNA is double-stranded with the two strands held together by hydrogen bonds. This is those bonds broken in a short section of the DNA. The opening between the strands is called a replication bubble. Lots going on here. I'll just list them. Upper left, SARS-CoV-2 AIDS, bacterial. We will be looking at the first fairly in-depth processes here. For every process you'll see, you'll need to know the steps in order. Think of them like stories. You wouldn't tell a child the story of Cinderella starting with trying on the glass slipper, right? These have a specific order too. Bacteria are prokaryotic, single-celled organisms. Viruses are not. They aren't made of cells. They can't reproduce on their own, but they can evolve. Are they alive? Under current definitions, review criteria for life from Chapter 1, they are not. Some scientists would like to change that. Note that it was stated in general terms, nucleic acid, not specifically DNA. That's because some viruses have RNA and not DNA. What kind of test would you do? Let's see what they did. Bacteriophage T2 was used in the Hershey and Chase experiment. It consists of DNA surrounded by protein, so it's a nice simple virus. The tail fibers attach to a bacterial cell, and some portion of the bacteriophage enters the cell to trigger production of new phages. At the time of the experiment, Hershey and Chase did not know whether the DNA or protein component entered the cells to initiate the process. Their experiment showed them which did and led to a new understanding of what DNA could do. The Hershey-Chase experiment. This was done in two separate experiments. In one, the protein was tagged with fluorescence, and in the other, it was the DNA that was tagged. The T2 bacteriophage were then allowed to attack E. coli cells. The cells were taken and spun down in a centrifuge. In the top experiment where the protein was tagged, you can see that the tagged protein remained outside the cells. In the bottom experiment, the tagged DNA was found inside the cells. That's what's in the pellet, since the cells are heavier than the liquid. Now they knew which part was entering the cells and causing changes. At the end of the chapter, we'll return to viruses, but for now, we'll just look at the basic life cycle of them. That's in quotes, because if they aren't alive, should we call it a life cycle? It is a replication cycle, but we are more familiar with the concept behind a life cycle, so tend to phrase it like that. In this case, the bacteriophage lands on the prokaryotic cell and injects its DNA. That DNA takes over the cell, causing it to stop doing what it normally does and use its resources to make more parts of viruses. The parts assemble and then destroy the cell on the way out of it. As we'll see later, there are multiple variations of this, but the basics are the same. The cell is lysed. Remember hydrolytic enzymes? Same root. To lyse is to break. Lysosome, same root. Which came first, the chicken or the egg? Are we just survival machines for our genes? DNA is a polynucleotide, a nucleic acid. Everyday functions vary by cell type, but all functions that relate to all aspects of the cell's life are controlled by the DNA. How food is acquired, processed, waste eliminated, how it protects itself, everything. Copies of DNA are what gets passed to the next generation, so all those directions. Do you remember what the abbreviations are for? Deoxyribose nucleic acid, ribose nucleic acid. Most of this is review. A polymer consists of monomers joined together. This is the macromolecule called nucleic acid. Both DNA and RNA are also polynucleotides. DNA has deoxyribose for a sugar, while RNA has ribose. RNA uses uracil instead of thymine, but both of these use adenine, guanine, and cytosine. 3 parts. A 5-carbon sugar. Remember the functional groups? Here's your phosphate group. And a nitrogenous base. One of 5 possibilities depending on the nucleic acid. What element do you think is in abundance on these? Nitrogen. This model has ribose for the sugar and uracil for the base, so is an RNA nucleotide. The structure of a DNA polynucleotide. This shows the specific complementary base pairing of the nucleotides. In DNA, adenine always bonds to thymine, cytosine always bonds to guanine. Note that the sugar phosphate backbone leaves the bases unaffected, so the 2 strands which make up DNA can bond at the bases. This model has deoxyribose as a sugar and thymine as a base, so is DNA. Remember, RNA has uracil in place of thymine. The bases can pair with one another by hydrogen bonding. Adenine and thymine can form 2 bonds. Guanine and cytosine can form 3 bonds. Purines always pair with pyrimidines to keep the distance between side chains constant, a 1-ring structure with a 2-ring structure, so the DNA is always the same distance apart. The bases must have the same number of hydrogen bonds. Thus, adenine always pairs with thymine and guanine always pairs with cytosine. Here's a memory aid. To serve apple pie, you must cut it. Cut, C-U-T, the pie, P-Y. Pyrimidines are C, cytosine, U, uracil, and T, thymine. The remaining are purines, adenine and guanine. A double helix is like a circular stairway, with the bases as the steps and the 2 sugar phosphate backbones as the side rails. The structure was discovered by Watson and Crick in 1953. Well, they get the credit for it. Rosalind Franklin is the one who gathered the data they used, but it was the 1950s, so she got no credit. You already know that the bases are held together by hydrogen bonds. Using base pairing to replicate DNA for cellular division. 1. The double strand is opened. 2. Complementary base pairs are added to each side to make 2 complete strands. Semi-conservative model means that each daughter strand has 1/2 of the parent strand. It is conserved in the daughter strand. You'll need to be careful with parent-daughter terminology. We did parent-daughter cells, now we have parent-daughter strands of DNA. It's not that any of these are female. The terms are to label which generation is which. There are very specific enzymes used during this process. I cover only a few. I should probably cover more, but I think it's confusing enough. Notice that under origin of replication, it says sites, plural. This process starts in multiple places on the chromosome, so it can go faster. The blue is the parent strand. The gray is the daughter strand. The arrows extending outward from the bubble are showing the directions the bubble grows. As the double helix is separated, new nucleotides are placed, complementary base pairing with the parent strand. The bubbles elongate as this happens and eventually merge together, separating the strands. DNA polymerase. Break the words down. The ASE at the end says it's an enzyme, and the rest tells you what it does. Enzyme that creates a DNA polymer. DNA ligase. Ligate or ligature means to join or join together. This one is an enzyme that joins short strands together. Visualize a line of people as nucleotides. And each left hand is a phosphate group, and each right hand is a base. The carbons on the sugar are important. Sugar 1 is the front of your right shoulder, 2 is the back of your right shoulder, 3 is the back of your left shoulder, 4 is the front, and 5 is the left hand holding the phosphate group. The line of people are all facing the same direction. You can place your phosphate group with the 5th prime, or 5th carbon, on the left shoulder with the 3' or 3rd carbon easily. The person behind you can do the same to you. New nucleotides, new people in line, must go to the end of the line. They cannot cut in front. The line grows from the 5', your left hand, to the 3', your left shoulder. If you look at the diagram, one side of the double helix goes in one direction, and the opposite side runs in the opposite direction. This is called anti-parallel. The sides of DNA run in opposite directions. This means that the new to daughter strands being built must also grow in opposite directions. This is half a replication bubble. One side can just build the daughter strand in continuously as the bubble grows using DNA polymerase. But the other side can't. It waits until a section is exposed, then jumps ahead a bit and builds short sections of polymer, again using DNA polymerase. These sections are called Okazaki fragments. DNA ligase then comes in and attaches the short sections together. This is a drawing of a full bubble. As you can see, since the bubble grows in two directions, continuous strands are on both sides, one on top and one on bottom. The same is true for the Okazaki fragments. So, how do we get the information out of the nucleus? The same way you get the information from the reference section home. You make a copy, of course. And yes, we need to build the proteins. This is where RNA comes in as a helper to DNA on getting the needed information where it's supposed to go. mRNA is the messenger RNA that carries the message, the directions to assemble amino acids. Transcribing DNA to RNA is still in the language of nucleic acids. Transcribing is to copy. Just make a copy. Translation is moving from the language of nucleic acids to the language of proteins. DNA is like a big cookbook. Each gene is a single recipe. When you ask a friend for a recipe, they give you a copy. That's transcription. When you make the recipe, you're translating it. There are markers in the genes to tell the cell where to start and stop. This is so you don't start in the middle of the beef stew recipe and stop in the middle of the chocolate cake recipe. Note the diagram. How is the mRNA leaving the nucleus? Yes, through those pores we mentioned in the earlier chapter. Each nucleotide has one base, so a triplet or codon of bases is 3 nucleotides. What are amino acids? The monomers of proteins. This will form the primary stage of a protein, the polypeptide. The codon chart comes in different forms. You may have a different type in your text. They all have a starting point for the first base of the codon, then for the second and third. On this one, first base is on the left. To find the codon GGC, first look on the left and find G, the first base. G is the bottom section. Then look across the top and find the second base, which is another G. This time it's on the right side. Third base on the right is where we need to find the base C. Notice that there are several. We need to find the one intersecting with base 1 and base 2. You'll see it codes for GLY. That is the amino acid it calls for. You'll see that many of the amino acids can be coded for by different codons. GLY has 4 different codons that can be used to get that amino acid. This is called a wobble. There can be minor mutations on some of these that will be completely neutral in effect. In eukaryotic organisms, all genes will start with AUG, which calls for the amino acid MET. They will all end with one of 3 stop codons, seen highlighted in the chart. On the right-hand diagram, a section of DNA is shown. The darker blue side of the template strand is the side to be copied. If you're colorblind, that's the one on top. Transcription forms the mRNA. The template strand is simply complementary base paired, using uracil in place of thymine to make the single-stranded RNA. Then the groups of 3 bases are read as a codon to determine the sequence of amino acids in the process called translation. Look at the codons above and see if you can find the amino acid on the chart. You will need to read a chart and do both transcription and translation on your exam. To replicate a strand of DNA on paper, you copy the entire thing. To transcribe it, you complementary base pair the template strand only, replacing thymine with uracil to make RNA. To translate, you use codons to find the amino acids and list them in the correct order. Note that we now have parent-daughter molecules. Also notice that the parental strand is black and green. The new sides of the daughter strand are red. You can see that each daughter strand has half a parental strand. Semi-conservative model. Do yours and then check it. AUG, GAC, GGA, UCG, AAG, CGC, UGC, UA, CGC, UAA. It's easier on our eyes if we had spaces between codons or draw vertical lines between them. tRNA is transfer RNA. It carries amino acids around and makes sure they get to the right spot by doing even more complementary base pairing. Since the mRNA has codons, the binding sites here are called anticodons. Notice that nothing is there to go with the stop codon. When one is reached, you must stop. Even if there are more nucleotides following it, you still stop. If you want more practice, just ask. You can post on the question discussion board or email me. mRNA is one we've already used, made by transcription. We've used tRNA, but didn't discuss where it came from. And now here's the third RNA, rRNA. There are 3 steps to transcription. Initiation, starting the process by RNA polymerase attaching. Since DNA polymerase is an enzyme that makes DNA polymers, you can see why RNA polymerase would be an enzyme that makes RNA polymers. Elongation, growing the strand by